Welcome to the deep dive. Today, we're tackling one of the toughest challenges in oncology, pancreatic ductal adenocarcinoma, or PDA.
It's a notoriously aggressive cancer, highly lethal.
And we're looking at some incredible research that points to a key reason why it's so aggressive. And, well, it involves the nervous system.
That's right. For years, we've known that PDAC is very good at something called neural invasion, NI for short. The cancer cells literally, grow into and along the nerves.
And that's not just a side effect, is it? It's a real indicator of how bad things are.
It's a huge independent prognostic factor. The more neural invasion you see, the worse the patient's overall survival. It's that direct.
Okay, so historically, how did we think that communication worked between the nerve and the cancer?
The focus was mostly on what we call paracrine signaling.
Explain that for us.
Paracrine signaling is sort of like sending out a general memo in an office. A cell releases a chemical signal, and it just drifts around in nearby cells. It's a bit diffuse.
Kind of like shouting into a crowded room and hoping the right person hears you.
Exactly. It's important, but it's a loose connection. What we're diving into today is something much more direct, much more deliberate.
So the cancer isn't just whispering to the nerve, it's installing a direct, hardwired connection.
Precisely. We're talking about the discovery of actual physical synapse-like connections between sensory nerves and cancer cells.
And this is happening in the pancreas, not the brain.
Outside the central nervous system entirely. This is a level of structural organization we really only expected to see in brain tumors.
So what's the hook here? The big, shocking finding.
The shocking part is that sensory neurons are forming what are being called pseudo-synaptic connections with these PDAC cells. The cancer is literally wiring itself into the body's nervous system to get ahead.
Okay, let's unpack that. A neuron-cancer pseudo-synapse. If I'm a scientist looking through a microscope, what am I actually seeing? What's the proof?
The proof is just stunning. It comes from transmission electron microscopy, Tim, on actual human PDAC samples. And what you see looks almost identical to a classic excitatory synapse in the brain.
The kind of connection that powers thought and movement.
The very same. You can see the nerve fiber, the axon, ending right up against the cancer cell. That nerve ending, the presynaptic bouton, is just packed with vesicles.
Vesicles being the little bubbles that carry messages.
Exactly. You see the large, dense core vesicles, which might carry neuropeptides. But the really crucial part is you see tons of small synaptic vesicles.
And those are for the fast messages.
The fast messages. They're the packages from molecular neurotransmitters like glutamate.
Okay, so the nerve is ready to send a signal. What about the cancer cell? Is it just a passive bystander?
Not at all. It's an active participant. On the cancer cell's membrane, right opposite that nerve ending, you see this dark, thickened area. It's called the postsynaptic density, or PSD.
I see. And what does that do?
That's the landing pad. It's a molecular scaffold where all the receptors are concentrated, ready to catch the signal the moment it's released. Finding that structure is the definitive proof.
So what is the signal? What's the chemical message being sent across this gap?
The signal is L-glutamate. This is the main excitatory neurotransmitter in the nervous system, and it's being sent by these peripheral sensory neurons that innervate the pancreas.
And the cancer cell has a special way of catching it.
A very special way. It's using a specific subunit of the NMDA receptor called GRIN-N2D.
So it's like the cancer has tuned its antenna to one very specific radio station.
That's a perfect analogy. The data shows PDA cells crank up the expression of two subunits, GRIN1 and GRIN-N2D. And that specific pairing creates a receptor that is hypersensitive to glutamate. It doesn't let go of the signal easily.
Let's connect this to the clinic. How does having more GRIN-N2D affect a patient?
It's a huge deal. In patients, elevated GRIN-N2D expression correlated directly with more aggressive tumors, specifically with the T and N stages.
Okay, let's quickly define those. T is for the size of the primary tumor, N is for spread to the lymph nodes.
Right, and high GRIN-N2D meant bigger tumors and more spread, and the single-cell data confirmed it. This wasn't happening in healthy cells. GRIN-N2D was selectively, jacked up only in the malignant cancer cells.
So let's do a quick recap here, about 5 minutes in. First, we have a new structure, a physical pseudosynapse between nerves and PDA cells.
Confirmed with TEM imaging. It's real.
Second, the signal is L-glutamate, and the cancer's receiver is a specialized receptor, GRIN2D.
A hypersensitive receiver.
And third, the clinical link. More GRFN2D means more aggressive cancer. So the big question is, how does receiving that glutamate signal actually make the cancer cell more dangerous?
Well, the functional outcome is really clear. Glutamate makes the cancer cells move. It dramatically boosts their ability to migrate and invade surrounding tissue.
How did they show that?
In the lab, they took PDAC cells and just added L-glutamate to them. The cells started moving and invading like crazy.
Did all PDAC cells do this?
No, and this is a key point. Only the neurofine cell lines responded, the ones that are already known to be attracted to nerves.
So this is a specialized skill. The other cell lines were essentially deaf to the signal.
Exactly. It proves this is a targeted mechanism, not a general effect.
Okay, so if GRIN2D is the on switch for this aggressive behavior, can you turn it off?
You can. They treated the cells with a specific GRIN2D blocker, a drug called UBP145, and it completely reversed the effect.
Yeah.
They also silenced the GRIN2D gene itself and got the same result. The aggression stopped.
So GRIN2D is absolutely essential for this. Now, we talked about the cancer cell being ready to receive the signal. Is it also telling the nerve to send a better signal?
It is. It's a two-way street. They found that the invading nerves had higher levels of the machinery needed to package and send glutamate, things like SLC17A6, the transporter, and VMP1, a release protein.
It's upgrading its own supply line.
It really is.
Yeah.
And on the cancer side, that postsynaptic scaffolding protein, DLG4, was also enriched. The whole connection gets stronger.
And they confirm this functionally, not just by looking at protein levels.
Yes, with calcium imaging. Glutamate triggers a huge influx of calcium into the PDA cells. But this is the critical part. It only happens when the cancer cells are physically touching the neurons.
Ah, so if the cancer cells are alone in a dish, the glutamate does nothing.
Absolutely nothing. They need that physical pseudo-synapse to be formed for the signal to work. It's a positional switch. If you're next to a nerve, you turn on this aggressive program.
That brings us back to GRIN2D itself. You said it was special.
Right. You can think of it as a conditional biosensor. It has these unique properties like a low magnesium block that make it incredibly sensitive to even small amounts of glutamate released right at that synapse.
So it's very efficient. It doesn't waste energy until the signal is right there at its doorstep.
Exactly. It's a perfectly evolved system for hijacking this neural signal.
Okay, let's follow the signal. Glutamate arrives, GRIN2D activates, and calcium floods in. How does that chemical flash translate into the cancer's long-term plan for proliferation?
So calcium is the trigger. That influx of calcium activates an enzyme called CMK-Phe.
CMK-Phe.
Right.
Yeah.
And CMK-Phe's job is to activate a very important transcription factor called CEB1. This is really the switch that starts rewriting the cell's genetic programming.
And that leads to a big epigenetic shift, right? It changes which genes are active.
A huge shift. CREB1 turns on the production of a protein called EZH2.
EZH2.
Yes, and the answer is S2. Now, EZH2 is a powerful gene repressor. Think of it as a librarian who goes around putting do not read signs on certain books.
And which critical safety manual is it putting that sign on?
It goes after a crucial tumor suppressor gene called CDKN2A, which you might know as P16.
P16, right. That's one of the cell's main emergency brakes for stopping uncontrolled growth.
Exactly. EZH2 silences P16. It takes the brakes off. And when the brakes are off, another transcription factor called E2F1 is liberated. It's free to act.
And this is where the loop comes in, I'm guessing. What does E2F1 do?
E2F1's job is to turn on genes for cell proliferation. But here's the really insidious part. One of the main genes it activates is EZH2.
Oh, wow. So it creates more of the very thing that set it free.
Precisely. It's a positive feed-forward loop. The glutamate signal starts the process removes the cell's own safety checks, and then that feeds back to make sure the whole aggressive system stays permanently on.
A self-sustaining cycle of aggression. Okay, let's recap again. We're about 12 minutes in. We've got the structure and now the engine. Glutamate hits GRIN2D. That leads to a molecular cascade that ends with the EZH2-E2F1 loop silencing the P16 tumor suppressor.
And that drives proliferation.
But how does this translate into the physical growth of more nerves into the tumor? How does it complete the loop?
That's the final piece of the puzzle. This sustained GRIN2D signaling also causes the cancer cells to release nerve growth factors.
Ah, so it's not just listening, it's calling for more connections.
It's sending out a beacon. When they knocked out GRIN2D, the expression of these neurotrophic factors plummeted. The most significant one was TGFA, transforming growth factor alpha.
And TGFA tells nerves to grow.
It screams at them to grow. TGFA, secreted by the cancer cell, enhances the growth of new axons from the sensory nerves, so the cycle is complete. GRIN2D signaling leads to more TGFA, which leads to more nerve growth, which supplies more glutamate, which further activates GRIN2D.
That is an incredibly elegant and terrifying mechanism. But how do we know this holds up in a living organism, not just in a petri dish?
The animal models were the ultimate validation. They implanted mice with PDA cells, and in one group, they had knocked out the Grin2 gene.
And the result?
A dramatic improvement in overall survival for the knockout mice. Their tumors grew slower, they were smaller, and they had far fewer metastases.
Just by deleting that one single receptor?
That one receptor. The knockout tumors were also less innervated, and they showed higher levels of apoptosis-programmed cell death. The cancer was literally killing itself off more often without that signal.
Did they definitively prove the tumor was reliant on the glutamate from those specific nerves?
They did a brilliant experiment for that. They used capsaicin, the stuff that makes chili peppers hot, to destroy the sensory neurons that supply the pancreas.
So they cut the wire.
They cut the wire and tumor growth slowed down. But here's the smoking gun. If they then gave the mice extra glutamate, it only restarted tumor growth in the mice whose cancer cells had intact GRIN2D.
I see. So if GRIN2D wasn't there, the extra glutamate did nothing.
Nothing at all. It proves the tumor's aggression is absolutely dependent on that specific nerve to GRIN2D connection.
And what about pain? PDAC is known for causing terrible pain. Is this axis involved?
It seems to be. The mice with intact GRIN2D and high levels of TGFA didn't just have bigger tumors. They also showed elevated mechanical pain sensitivity.
So this pseudo-synapse isn't just for growth, it's likely contributing to the patient's pain.
It's very strongly implicated.
This is all pointing to a clear therapeutic target. But targeting glutamate receptors in the brain is famously difficult because of side effects. How do you get around that?
Well, this is where the new understanding is so powerful. We now see two separate NMDR pathways. An older one, involving a subunit called GRIN2AB, seems to be for the cancer stimulating itself. But this new GRIN2D pathway is specifically for receiving signals from the nerve.
At that physical connection point.
Exactly. So the therapeutic potential is enormous. You can design a GRIN2D inhibitor that does not cross the blood-brain barrier.
A non-BBB penetrating drug.
Yes. Because these pseudo-synapses are peripheral, you can target them locally without messing with all the critical glutamate signaling in the brain. That could mean a much wider, much safer therapeutic window.
What an incredible story. The cancer cell becomes this neurosavvy predator, wiring itself into the body for a growth advantage. But because that wiring is peripheral, we might be able to cut it without causing collateral damage.
I think this discovery really shifts the paradigm for cancer neuroscience. It suggests that oncological therapies designed specifically to dismantle these peripheral connections could be a major new frontier for treating PDAC.
So here's a final thought for you to take away. If these localized, non-BBB penetrating GRIN2D inhibitors work for pancreatic cancer, could similar pseudosynapse mechanisms be at play in other cancers known for high neural invasion? Think about colon cancer, prostate, or even certain breast cancers. Could this discovery unlock a whole new class of therapies across oncology, all based on the simple idea of cutting the cancer's private communication line to the nervous system? That's something to consider. Thank you for diving deep with us.
My pleasure.